Nope. All right. Great. So first is the science of COVID-19. And here I'm delighted to introduce Dr. Jay Grella. Those of you who know my last name will notice that we have the same last name. So Dr. Grella is also my father. And I was lucky enough to get him to agree to give us a lecture here. So Dr. Jay Grella is Emeritus Professor of Biochemistry at UCLA. He's known for basic studies that define the critical biochemical steps that are associated with transcription initiation. And his studies are unique in integrating concepts across the biological kingdom, from bacteria to yeast to man. And he's been at many prestigious places, Yale, Harvard, MIT, and the Pasteur Institute. And if you don't understand what those biology words mean, basically it means that he knows a ton about RNA, and this is an RNA virus. And I think the relevance of his expertise will become clear during his presentation. And it's also relevant to note that he is currently writing a blog that interprets a lot of the science behind COVID-19 for a lay audience. And he's drawing from that for this lecture. And you see the link on the slide here. So, Dad, thank you for being here with us.
Oh, it's a pleasure. Okay, so shall I upload my slides and get started?
Yep.
All right. Okay, are we all set now?
We're here. We're good. I hear you.
Okay, so today you get something just a little bit different, namely biotechnology. And there are going to be two parts to this talk. The longer part will be the origin of the virus very briefly, and mostly the biotech tools that are able to map the virus as it spread around the world from Wuhan. And then the second part of the talk, which will be shorter, we'll talk about the tests that are used for learning different things about COVID-19. And hopefully at the end, I've got 3 slides for how one designs and tests vaccines. So just get started with this now. So COVID-19 is a virus that in nature exists, its reservoir in nature is in bats. That's pretty common for RNA viruses. But this virus cannot spread directly from bats to humans. Instead, it needs an intermediary creature in which it can mix its RNA with the virus from that creature. And this first happened in 2003, at least that was the first documented case, came out of a live animal market in Guangdong province in China. At the time, it was predicted that it would be some years and then a second virus had emerged somewhere. And that second virus, sadly, has now emerged. That came from a different wild animal market, this one in Wuhan. The intermediary creature was not a civet like it was in the other market, but is now a pangolin. So let's just talk a little bit about the biology of how COVID-19 spreads. There's been a little bit of understanding as well as misunderstanding about this. So one thing for sure is that the origin of almost every case of COVID-19 is exhalation, that is coughing or sneezing or singing or even breathing hard. Now the virus lives in your respiratory system, which is a wet place, and it comes out, as you can see in this display, in various little wet particles. And the science of these particles is totally non-controversial. And I'm going to just show you how these particles behave in the next few slides. So within 3 to 6 feet of a sneeze or cough, you're going to find what chemists think of as fairly large droplets. Now, the bad news about these droplets is that because they're fairly large, they have a heavy load of virus. So you wouldn't want to be right next to this exhalation. The good news is that because they're heavy, they fall down to the ground fairly quickly, and they do start to lose their infectivity. So this basically is the origin of the 6-foot rule. These large particles droplets rarely travel more than 6 feet. So you're unlikely to get hit by the direct droplets if you're more than 6 feet away. Now, beyond 2 to 3 or 4 feet, the droplets turn into aerosols. And these aerosols are much smaller, and therefore they have a much lower content of virus, but there are an awful lot of them. Now, these aerosols are not dangerous in terms of spreading infection if you're outside, because basically they just float. Just imagine some dust motes floating around. These act similarly. They'll just disperse outdoors, and they're of no worry. But indoors, they're potentially a very big problem. They don't fall to the ground quickly. They linger in the air for at least minutes. Most likely, they linger in the air for many hours. And under these circumstances, the 6-foot rule is no longer remotely relevant. And I'll show you that on one more slide here. So I just took this guy away. He left the room, but you can see what he left behind. He left behind this virus mist that is now floating in the air, containing infectious particles. And if you're indoors, that mist is going to linger from minutes to hours and is a potential source of infection. And there's been a little bit of misunderstanding about this, but this should not be controversial. All right, now let's move to the biotechnology part of this. You've probably all seen some version of this slide. This is a cutaway of COVID-19. On the outside, you have this famous spike protein shown in blue. And in the inside, this folded up thing is the famous RNA. Now, if we're going to detect COVID-19 with biotechnology, we will be targeting either the spike protein or the RNA. Now, to target the spike protein, the technology that's used is called immunochemistry, which sounds quite daunting, and quite frankly, it is pretty daunting. And so I'm not really going to give you anything more than the most superficial view of immunochemistry and the spike protein. The RNA, that of course is really the heart of the beast. All the information for the infectivity, the reproduction, the entire life cycle of the virus is contained solely in the RNA. Now, the jargon that you will hear and that I will use today is we'll be talking about the sequence of the RNA. So here's what I mean by that. This RNA is one linear chain, and the chain has in it about 30,000 links, and each link is given a different letter. The sequence is just the order of letters. So if you look down on the bottom right, you'll see a sequence of letters, A, G, C, and so forth, 30,000 of them. The original virus that came out of Wuhan had one sequence 30,000 long. So that's what I mean when I say sequence the RNA. Now, this RNA is changing a little bit as it moves around the world. And so when I say we sequence the RNA from a patient, we mean, is it the same as what came out of Wuhan, or has it changed slightly? Now, there's a challenge here because COVID-19 is part of the coronavirus family. And these coronaviruses are all quite similar one to another. So if we're going to track coronavirus, we can't be tracking, I don't know, a bat virus, for example. We have to use the part of COVID-19 that is unique to humans, so we avoid counting viruses from other organisms. Fortunately, we know how to do that, and the main tool that is used, aside from learning the sequence of the RNA, is a so-called sequence algorithm, sequence alignment algorithm. So I've shown the initial output from a sequence alignment algorithm in this slide. So here's how it works. Each row here represents the RNA sequence of a coronavirus from a different organism. The SARS one in the middle is a human coronavirus. So you input all these RNA sequences. The algorithm then arranges them into families. So up at the top, you see I've bracketed, 5 virus sequences, and they constitute a small family. And you can tell they constitute a family by, if you look in the middle, you'll see some dashes. Those dashes are where letters are missing. That is, these viruses are part of a family that has a slightly different sequence, one that contains fewer RNA bases or letters in the middle. Now, this is probably a very full arrangement of a family. These algorithms are very powerful. They can handle hundreds of thousands of RNA sequences and align them in very sophisticated ways. So in the initial alignment tool, the output looks something like this. I've selected some organisms. What you see here are the identities of the sequences of these organisms to the human COVID-19. So look at that. 96% of the genome of the bat coronavirus is identical to the sequence of COVID-19. I said it contained pangolin sequences, 91% identity between pangolin and COVID-19. The 2003 human SARS virus, that's 80% identical to the current virus. And probably not too many of you know that there is some version of the common cold, a minor population of colds are caused by coronaviruses. And those viruses are 50% identical to COVID-19. But of course, 96% is not 100%. So we do have unique sequences in COVID-19 that allow us to not only track it, but distinguish it from other viruses in the family. Now, in addition, the tracking will be aided by mutations that will come into the virus. As the virus moves around the world, we'll be able to follow these mutations and thus follow the virus. Let's have a look at those now. Okay, now I'm going to explain how these mutations form. And in my blog, I call these typos. Now, molecular biologists don't call these typos. They call them point mutations or substitutions. And I lifted this picture from a textbook. It's not really for the reproduction of RNA. It's really for the reproduction of DNA and how mutations go in, but that shouldn't matter today. So if you look along the color coding on the bottom part of this colorful picture, you'll see a series of letters corresponding to the sequence: green A, orange C, purple T, and so forth. And the aim here is to preserve the information inside the cell, and there's a molecular machine that does that. And this is an amazing machine. It only makes a mistake inserting the wrong partner base one time in roughly 1 million. So every time you see a C on the bottom, little orange C, the machine knows to put in this G on the top. And it's done that very well until it gets to the arrow that I placed in there, where it's attempting to put in a green A instead of the proper G. And this is a rare event. It only occurs maybe one time in 1 million copies, but copying events, that is. But because there's 30,000 bases in a genome, and because you end up with millions, billions, and trillions of viruses from infections, eventually the population of viruses accumulates some of these mistakes. Now, I know mutant is a scary word from the movies, but in fact, very, very few, if any, of these mutations are harmful to humans. A lot of these are harmful to the virus. They make the virus less infectious. The majority of these mutations are silent. They do nothing whatsoever to the infectivity of the virus. Has there ever been a mutation that makes the virus more infectious? There's been discussion that maybe one exists, but even that one is controversial. So nearly all these mutations are just randomly occurring as the virus goes from individual to individual, and errors accumulate during the reproduction. Now, as of July 2020, the typical virus differed in eight positions among the 30,000, 8 random positions. And this comes from the virus moving from person to person during the 6 months or so. And we're going to use, we being scientists, will use the nature of these mutations to track the virus, because if only 8 random ones are induced out of 30,000, well, if I've got one with those 8 and you've got one with those 8, it's very likely that one of us gave it to the other. Now, if somebody else turns out to have only 7 of those 8, probably what happened was they had the 7 and they passed it to me or you, and we introduced the 8. And if you have enough of these examples and a good enough algorithm, you can actually track the virus by these silent mutations as it moves from place to place. And I'm going to tell you about that in the next few slides. All right, this looks complicated, and it can be, but it's really not so bad. This is the way things looked in, I think, August, just a month or two ago. What you see here are 40,000 dots, and each dot represents a patient who donated a sample of RNA, and that RNA was subject to sequence analysis and placed in the alignment algorithm that I discussed with you just a few minutes ago. As I said, the algorithm grouped them into families that are closely related to each other in RNA sequence. There's obviously a dark blue family and a green family and so forth. Now, these families arose with time by mutation. And so as we go from left to right, this is basically a time scale. January on the left and August on the right. And so the color coding actually refers to, I think in this case, it's the family of virus. Color coding will change in a minute. Okay, so let's just interpret this just a little bit more to make sure you understand it. Okay, so I said it's temporal from left to right. So the arrow on the left represents the earliest events. These are the ones occurring in January. The version of the virus that occurred in January was the one that arose in Wuhan, and that's represented by orange dots. So you see above January, there are essentially only orange dots. Now each dot is also geotagged, and so we know that each patient giving rise to a dot was from China. And of course, there's no surprise because that's where the virus originated. Now, as the virus spread from left to right, you can see we'll make our way now to what it looks like in July. So over on the right in July, you can see that in terms of viruses that are related to the original one and are in China, there are really very few cases. China has the epidemic significantly under control. But of course, it's spread all over the world. And now the dark blue dots are strains that exist largely in Europe, but elsewhere in the world. The green family and the light blue family, I mean, these things are everywhere now. And I did not take the trouble to write out the geotags for you. But basically, what I'm showing you is a display illustrating how the virus has formed families as it spread along the world. Now, of course, each dot represents a person. So we actually know not only where these have spread, but who spread them. And more sophisticated algorithms can take advantage of the kinds of silent mutations that I told you about a few moments ago to attempt to track the spread in both space and time. And we'll have a look at that now. So this is really molecular genealogy here, right? It's basically saying, okay, who had the virus first? Who did they give it to? Where did it go? And what happened next? So the yellow ball in the middle represents, this is actually, the color coding is a little different here. Sorry about that. It's not my fault though. The color coding in the middle, yellow indicates cases that occurred early on in China. And each line represents the transmission of a virus, which you can tell from its sequence to another country. And the countries are all color-coded there. The darkest blue ones are individual spreads from wherever to the United States. And of course, it's spread everywhere now. And I want to look more closely at one line, the dark gray line, that was originally emphasized by the authors of this study. So look at that dark gray line. You can see that the virus traveled from China, yellow, to Munich in Europe, light blue, from Munich to Italy, from Italy back to Germany, which of course where Munich is located, and also to Mexico. Now, what I want to emphasize here is that all of this is done without interviewing a single person, right? This is all biotechnology tracking of the spread of the virus. Now, the hope was that you could use this kind of biotech tracking as a supplement as well as a complement to contact tracing. And so this dark gray line was subject to human contact tracing in case we could learn how the virus actually spread from China to Europe. This dark gray line represents the earliest case of spread that was in the database. So let's look at what happened in contact tracing. Okay, so what we have here is the patients represented by the dots, or the individuals, were interviewed. And it turned out the original employee, the original person in China was actually from Shanghai, not from Wuhan, but her parents were from Wuhan. And so in very early January, this woman had received a visit from her parents in Wuhan. Apparently, her parents were infected, but they didn't feel very sick, apparently, and spread the virus to her. Now, of course, at this time, it was not known that this virus existed, at least it was not known outside of China. So she felt fine, or fine enough, to agree to go back to Munich for a few days to company headquarters for a short meeting. She went back to Munich for, I think, 3 days. During one of these days, she had a lunch meeting with a few colleagues in a small conference room. At that lunch meeting, she spread the virus to one of her colleagues. This now being late January in Germany, the colleague decided it was time for a nice Italian vacation. He went down to Italy. The infection spread to Italy using his version of the virus. These are very early days in the spread to Europe. He, of course, went back to Germany and helped further spread the virus there. The employee, of course, went back to Shanghai and helped spread the virus there. From Italy, the virus then spread by somebody taking a vacation to Mexico. And so what I'm trying to show you here is that you can do biotech tracing without actually interviewing anybody. And you can actually go back and interview the individuals and reconstruct retrospectively how it is the virus spread.
There was a question about what it means when a dot has multiple colors in it.
When a dot has multiple colors in it, it probably means that that person has 2 versions of the virus inside them, depending on which graph you're asking about. It probably means the person has 2 versions of the virus inside them. One version they might have gotten originally, and the second version probably came up by mutation in their own body. Is that okay?
Yep, thanks.
So I'm going to move to the second part of the talk here, which has to do with testing and immunity. Now, this is the first part of the talk that you're going to have to pay close attention to the biology. And if you get the biology of this slide, you'll be fine for the rest of the talk. So a week or 2 after a virus infects you, your body makes antibodies. And these antibodies are represented here by these little green Ys. The antibodies are actually Y-shaped. And the antibodies then attack the virus and mark it for destruction. They also attack the virus infected cells in your body and mark them for destruction. Now, what you need to know here is that the way they do this is by recognizing some part of the virus. And the part of the virus that they recognize is called an antigen, and you need to remember that word. So an antibody binds an antigen, but it's your body that determines which antigens to use. And so your body actually makes hundreds or thousands of different antibodies, which can recognize hundreds or thousands of different little bits on COVID-19. What all these things have in common is that the antigen binds the antibody. That's really what has to be remembered here. All right, now let's back off from this a little bit. The mere presence of antibody in you means that you had COVID-19. Now, it doesn't mean you have it now, because the antibodies hang around for quite a while after you've been infected. But if you have antibodies, it means you have the virus now or you had it once. And so what we're going to do now is to look at some of the tests that are applied during this pandemic. And we'll have a look at the antibody test, which is the test that says whether or not you have had COVID-19. Okay, so there's 3 different tests that are being applied during the pandemic. Let's look at this antibody test first. As I said, the antibody test tells you whether you have a past infection. It could be a current infection also. Where are these tests done? They can be done in central laboratories, but antibody tests can also be done at point of care. That means when you go to a doctor's office, mostly. The antibody tests are really very fast tests, and they've received a lot of bad press, but they're really, at this point, quite reliable. And they're very cheap. It really is only maybe $3 or $4 to do one of these tests. Antibody tests exist that you can do at home. They just have not yet been FDA approved. If and when they are approved, they'll be just like a home pregnancy test. They'll almost the same technology, actually. So that's a test for whether you have ever been infected with COVID-19, but how about whether you have a current infection? And those are the 2 tests shown above, the RNA test and the antigen test. The antigen in this case means spike protein. Okay, so when I say antigen here, I mean spike protein. Now, if you have the RNA or the spike protein in you, it's a pretty good bet you've got a virus, and so you'll be considered positive if these tests turn out to be positive. Both of these tests can be done in central labs, but the RNA test cannot be done at point of care. The machines and technology is just too complicated. The possibility of contamination is too great. It can only be done centrally. But the antigen test can be used at point of care. And in fact, it already is. Many assisted living facilities and nursing homes have now adopted the antigen test for weekly testing, and they just go around and they test all their workers and in some cases all their residents. This test can be done on site in a few minutes. It's as reliable as the RNA test, and instead of costing hundreds of dollars, it only costs $5. It will be available eventually. Well, it's available to not for the general public, but it works as a home kit as well. The RNA test is the most famous one, but it will never be available as a home test, and it's never going to be really cheap. So the antigen test should take over eventually. Now, the technology behind these tests is really it's complicated, and I really don't have time to discuss the details of it. I'll give you a superficial idea from this by talking about the antibody test, the one we just discussed. So I want you to imagine that there's 96 people. I picked 96 because of the kind of robot that's shown in this slide. 96 people who want to undergo the antibody test to see if they've ever been exposed to COVID-19. So they donate samples, 96 samples that are worked up by a robot. Those 96 samples are placed in a multi-well plate that you can see on the top of this machine on the right. This is basically just a piece of thick plastic in which has been molded, in this case, 96 wells. And each well will then get a sample worked up from a different patient. Now, to tell whether that antibody is present, biochemists have designed antigens that bind that antibody, that we can purify those antigens in the lab and modify them in a way such that if they bind an antibody, a series of chemical reactions occur, which eventually leads to light emission, that is, the sample turns a color. So these 96 samples, each in a well, will have this reagent containing this color-forming chemical that I just described to you. They'll be injected by these pipettes, as you see here. If a person has an antibody, it will bind this reagent. Color will be produced, and inside this machine, there is basically a color detector. And so for each well, there's a color detector under it, and you will be able to know which, if any, of those 96 people have been exposed to COVID-19. 96 is a small number. Now they have these plates that hold on the order of 1,000 or more little wells. So this is really highly automated. I can't go over the chemistry done for the RNA tests involving RNA or antigen, but it's basically done in the same kind of setup. The only thing that's really different is the reagent. That's all. Okay, so I've finished what I can tell you about testing in the time available. And now in the last 3 slides, I'm going to tell you something about a topic that is in the news a lot these days, for better or for worse, and that is vaccine design. All right, here's the same picture again with antigens bound to antibodies. The antibodies, if they bind, are marking the virus for destruction. Now, antibodies are great, but the problem is that it takes 1 to 2 weeks for your body to produce antibodies after you've been exposed to a virus, any virus. So those 1 to 2 weeks are really the most dangerous period for you because you have other parts of your immune system assisting in destroying the virus. But the most powerful creature is the antibody. And it takes 1 to 2 weeks for your body to create antibodies, because to create these antibodies, it has to seek out these little bits of the coronavirus and learn how to bind them and then create proper antibodies. An amazing process, really. Now, as these antibodies are made, as you get into week 2 and week 3 and week 4, the antibodies are stored in memory cells. And perhaps more importantly, inside these memory cells, the information for making more of these antibodies is also contained. So what this means is the first time you get an infection, it's hard because it takes weeks for the antibodies to appear. But if you get that infection again, 8 months later, those memory cells will produce the correct antibodies quickly in 1 or 2 days. And they'll basically destroy the virus before it can gain a foothold again. That basically is the definition of immunity. That's why you're immune to something. Your body has learned to make memory cells storing quickly deployed antibodies. So what's the goal of vaccination? It's basically to induce the body to make memory cells that will work against COVID-19. All vaccines have this in common. Now, how do you do this? Here we move a little bit from science to a mixture of science, art, and a lot of luck. Basically, what you have to do is to choose an antigen and deliver the antigen. But you have so many choices for choosing the actual antigen and choosing how it's delivered. Because remember, the whole spike protein is really too big to be an antigen. It's just a tiny part of it. Which part to choose? Let's have a closer look at the spike protein on this slide. So this picture down on the lower left is what that red blob from the previous slide really looks like in close-up. It's an immensely complicated surface. And this surface could contain thousands of different antigens. Which little bit is the best bit to use? You have people in every vaccine company all over the world staring at this picture, saying, which part of it do we want to choose for an antigen? Once they've made a choice, they isolate that little bit from the spike protein, they produce large amounts of it, and that's the basis for their vaccine. If they choose correctly, they'll have a great vaccine. If they choose poorly, the vaccine won't work. So they might choose the red part, they might choose the green part. There are mild reasons for choosing green over red or red over green, but it's still as much of an art as it is of science. Then you have the question of how to deliver the antigen. How much of this little red or green bit do you inject? Too little and it won't work. Too much and you might have side effects. How do you deliver it? All at once so you get a huge response? Or do you have a time release kind of delivery? It's very hard to predict these things. And on my last slide, I'll show you these questions in just slightly more detail. So what you've heard about vaccines is, are they safe and effective? Here's the story about a safe and effective vaccine. It all has to do, well, the effective part has to do with how tightly the antibody binds the antigen that you happen to choose for your vaccine. If you chose well or luckily, you'll have a tight antibody attachment and you'll have excellent immunity. If you chose poorly or unluckily, you'll have weak antibody attachment, and therefore these viruses will not be well targeted for destruction, and you'll have poor immunity. How about side effects? Side effects are completely unpredictable. There's no scientific knowledge at all that allows us to predict whether there will be side effects. The side effects come about for, let's say, for example, you chose the green bit as your antigen. If just by bad luck, that green bit had a little part of it that looked like a piece of protein on the surface of, oh, I don't know, say your kidneys, then the antibodies would go and seek out that little piece of protein in your kidneys. And you could start to get destruction of your kidney cells. And since the interaction between that antigen and this antibody might be kind of weak, it might actually take months for the kidney damage to show up. And that's why it takes many months or years under normal circumstances to test vaccines. How many months will we allow? This is not a decision of scientists. This is a decision of politicians. So stay tuned. That's it. So I'm basically done.
Okay, thank you. That was fascinating. And I have a bunch of questions, but I am not going to take away from my students' ability to ask questions since I can do that anytime. So if you have questions, please raise your hands or type into the chat box. So let me read this one out. Sam says,
you read it because it appeared here.
Yeah, I'll read it. Is a single vaccine applicable for different